Hi, my name is Yerin Kim. I'm your today's host. I'm a recent graduate from Bioinformatics program at University of British Columbia, and I'll be your host today for BAMBUG seminar. Please silence your phone if you haven't already, and this event is being recorded. Starting from 11, it'll be followed by introductory training talk and also feature talk after that. BAMBUG organization committee is consists of Dr. William Seo, Dr. Emily, and Dr. Frost Hawk, and also student volunteers by Irana, Carolyn, Jonathan, Fatmeth, Parham, Bardia, and also me, Yerin Kim. We have upcoming BAMBUG event in February and also March to be announced. And you can also subscribe to BAMBUG if you want to by email with the following email address. And you can also subscribe to our calendar if you want to. Please suggest a speaker you can think of for the next session or next season if you want to. And BAMBUG is sponsored by Graduate Studies in Bioinformatics at University of British Columbia, Genome, British Columbia, SFU, University of British Columbia, Michael Smith Laboratories, also Langer College, Stem Cell Technologies, and BAMBUG is affiliated with ISCB and also sister bugs, Mombug, and Torbug. If you have any questions, please leave it on the chat, or you can just directly ask after the talk. So for the introductory student talk, it will be Rituparna Banerjee and a PhD candidate in Bioinformatics program, jointly supervised by Daniel Coombs and Matthew Pinnell. And the title of the talk is Exploring B-cell repertoire revolution post-vaccination using mathematical modeling and phylogenetic trees, and also followed by a feature talk by Dr. Graeme Koelwyn. He's an assistant professor with the Department of Health Sciences at SFU, Canada Research Chair Tier number 2 in Public Health Omics and Exercise and Disease. And the title is Inflammatory Memory, Using Omics Tools to Illuminate New Pathology in Health Disease. Okay, for introductory student talk, Rituparna, can you please share your screen? I'll stop my share.
Should I start?
Yes, please go ahead.
Hi, everyone. My name is Rituparna, and I'm a PhD student in the Bioinformatics program at UBC, co-supervised by Dan Coombs and Matt Pinnell. Today, I'll be talking about one of my projects, which is trying to understand how B-cell repertoires evolve post-vaccination using mathematical modeling and phylogenetic trees. Before I dive into my project, I want to acknowledge that I work on the traditional, ancestral, and unceded territory of the Musqueam people. Okay, so we all know how important vaccines are, and in order to reap full benefits of vaccinations, these need to be administered such that in a very thoughtful manner, with keeping in mind certain factors like the gap between doses, what the doses consist of, etc. So I am trying to look at broadly two different vaccination systems. One is heterologous vaccination, where the first dose and the booster doses consist of the same strain, and heterologous vaccination, where the two doses consist of different strains. Now, the main goal of my project, as I mentioned before, is to try to understand how immune response evolves in these vaccination scenarios. So, oh, sorry. So I'm trying to build mathematical models of immune response generation after each dose, and also trying to use empirical data, try to build phylogenetic trees of the B cell repertoires, and trying to gain insights. Now, specifically, I want to ask these questions. Firstly, how does the immune response vary between two doses in each vaccination system? How does the overall immune response vary between the two systems? I also want to see how well my model captures the evolutionary signatures present in empirical data, and also vary time between vaccine doses to see how that influences diversity of the repertoire. Now, I want to look at differences in immune response by looking at breadth of immune response, which means what are the different kinds of immune cells responding, and also strength, where I want to check what is there a difference in abundance of the responding immune cells. So this project can help inform vaccination strategies, as in what kind of system generates a more robust response in case of particular infection, or what should be the ideal time gap between doses in each vaccination system. So I have been talking about immune response, but in the context of my project, what I mean are B cells. B cells are immune cells that produce antibodies, and each individual has a unique repertoire of B cells in the body. Now, these cells evolve after exposure to a vaccine or infection, and thus helps to maintain a diverse repertoire in the body. B cell response is generated through two different pathways, the extrafollicular and the germinal center pathways. Now, in case of first exposure, which can be the first vaccine dose or first infection, dendritic cells carry the antigen to the B cell follicle, where the naive B cell is present, and these dendritic cells present these antigen to the naive B cells, and with help from T follicular helper cells, activated B cells are produced. Now, some of these activated B cells may have a higher affinity and may want to take the extrafollicular pathway, which would produce response by forming plasmablasts. This is a short-term response. Antibodies are produced, but the response dies off after 3 to 5 days. In the meantime, B cells of intermediate affinity may enter the germinal center, undergo rigorous rounds of proliferation, mutation, and selection to produce a more directed response in the form of plasma cells, which secrete antibodies, and memory B cells, which help to keep a memory of this present exposure for future re-exposures. In case of re-exposure, which can be a booster dose or a reinfection, the dendritic cells are already presenting the antigen to the naive B cells, as I mentioned before. But now there's also memory B cells from previous exposure. With the help of three T follicular helper cells, different categories of activated cells may be produced. Memory B cells of high affinity, if the strains from previous exposure and this one are antigenically close, may take the extrafollicular pathway and produce response by forming plasmablasts. Memory B cells of intermediate affinity and naive B cells of intermediate affinity will want to enter the germinal center to produce a more directed response. And studies have shown that there is competition between memory B cells and naive B cells to enter the germinal center. Now, all the studies so far have mainly focused on germinal center reaction as a mechanism of immune response generation. But I want to combine both the methods of immune response generation, the EF and the germinal center pathway, to see how that influences B cell profiles in these two vaccination scenarios. And I also want to see whether that leaves an evolutionary signature on the phylogenetic trees of B cell families. So in order to do that, I start by simulating a naive B cell repertoire, which mainly consists of bit strings, each bit string representing a B cell strain and another bit string to represent the vaccine dose. The reason I've chosen bit strings is because it can be a good representation of the high dimensional antigen antibody sequence space. Using this simulated repertoire, I run my model for first dose as well as for booster dose. And then at the end of, after running the model, the simulated repertoires are used to build phylogenetic trees. And next, I use some tree metrics to try to compare between the trees. Now, the model that I'm building is a dynamical mechanistic population model. It includes a two-dose vaccine regimen, each vaccine consisting of just one antigen. And it includes both the EF and the GC pathways. Now, I also use matching score as a proxy for affinity. And it is calculated by subtracting the number of mismatched sites from the total length of the bit string. So this is the model of my first vaccine dose, where I have the simulated naive B cell repertoire and the antigen. And upon interaction, they produce the activated B cell repertoire, which based on affinity takes either the extrafollicular or the germinal center pathway. This is the model of the second vaccine dose, where apart from the simulated naive B cells, there is also memory B cell population from the previous dose. And both of these interact and can take either of the pathways, depending on their affinity. So after I have run my models and I have my simulated repertoires out at the end of vaccination, I try to build phylogenetic trees and try to compare them using tree metrics. So one of the tree metrics that I plan to use is normalized Collis index. This is a tree metric used to describe tree balance. I also plan to use a few other tree metrics like gamma index, phylogenetic diversity versus mean pairwise distance, and phylogenetic abundance evenness. So these are some of my very preliminary results. This is a work in progress. And so these plots are more of a proof of concept to show that my model is behaving in the way I wanted to. So I started this toy simulation with 12 bit strings, each 6 bits long. And the same strain was used for first dose as well as the second dose. So during the first dose, once antibodies were generated, the number of vaccine particles started getting cleared out rapidly. And after the second dose was administered, since memory was already present, the vaccine particles started clearing out faster from the beginning itself. So next, I tried to also calculate Collis index. I tried to run 3 more simulations with 12 bit strings, each 6 bits long, but not necessarily the same repertoire. And I found varying trends in the Collis index. So it is kind of inconclusive at the moment. I was expecting the Collis index values to be higher after the second dose because I would expect some of the lineages to evolve faster than the others. However, I will need to run, I guess, I need to run as part of my next steps more number of bit strings with longer sizes and see whether I observe some trends. And also, I need to calculate other tree metrics and see whether I can answer my questions. I also, as I mentioned before, I intend to use empirical data and and build phylogenetic trees using maximum likelihood algorithms. And finally, I intend to extend the model to include 3 doses of vaccination and vaccine doses containing 2 or more antigens, and also intend to extend it to infection. So lastly, I want to thank my supervisors, Dan and Matt, for their guidance and all the members of the 2 labs. and my supervisory committee members, Eileen and Jeff, for their valuable inputs, and also Yeshwan, who helped me get a better idea. Thank you for listening.
Thank you. Thank you for the talk. I really appreciate it. I have one question. I was wondering how many patients were under the treatment, the vaccine. Can you please tell me the size?
So the patients were part of the empirical data, and for the data set that I found for homologous vaccination, it was 7 patients. Like the sizes weren't big, and I think it has
Mm-hmm.
to do with, like keeping track of patients over time. So,
Mm-hmm.
so the homologous vaccination one had 7 patients, and the heterologous vaccination one had 5 patients. So not a lot. Yeah.
Okay. Do you have more plan to track them and do more treatment with different dosages or something?
Um, Actually, so my main focus is on trying to understand the mechanisms. I do
Mm-hmm.
want to like I will keep looking for data sets with bigger, cohort size, but then and also with different doses. But that is like something for the future, actually. Yeah.
Oh, yeah, for sure. Yeah, I understand what you're trying to do.
Yeah.
Okay. Yeah, that sounds good. All right. Anyone in the audience have any
Oh, fine.
questions? Um, okay. Uh, if
Sorry, I have
uh,
a quick question.
okay, sure.
Hi, Rituparna, great work.
I
I'm wondering if uh, sorry, you had those plots of the direction of balance and that it was different
Yes.
than what you'd expect.
Yeah. This
Yeah,
one or
the red and blue one.
yeah, cortisol.
So first, I just wanted to understand if you're using balance, like imbalance, as a metric for like selection, selective pressure. Is
Yeah,
that
yeah,
the case?
yes.
Wonderful. And then did you observe this pattern both in your simulation and empirical data or just in simulation?
No, this is just in simulation, and also like this is a toy simulation. So with like really less number, like I started with 12 bit strings, and each were just 6 bits long. So I observed these trends for those.
I see. But
Yeah.
in real data, you do see the imbalance, right? After this, like,
Yeah.
interesting. Okay, thank you.
I mean, I haven't tried it with real data yet. That is something I want to do, but I want to also run it first, my simulations with longer bit strings. And then, yeah, I intend to check real data too. Yes.
Makes sense. I'm just wondering if, like, at the initial stage, there's just more diversification, and so you wouldn't see that imbalance until later when uh, there's the actual selection and cutting off of the less useful versions.
Versions, yeah. Yeah, that's true. Like, I would also expect that. Um, but yeah, like, I don't have, like any trends that I observed yet.
All right. Okay, let's move on to our feature talk by Dr. Graeme Koelwyn. Can you please share your screen? Yep, I can see your screen. Uh, by the way, you're muted.
Sorry, when I opened it up, it kind of bump stuff. So you can hear me and see me now, right?
Yes.
Awesome. All right. Yeah, thanks so much for the invite to talk. Yeah, my name is Graeme Koelwyn. I'm a principal investigator at the Center for Heart-Lung Innovation at St. Paul's Hospital. So our center primarily deals with clinical translational science in the fields of heart, lung, and critical care disease. So I was brought in there in, I guess, at the end of 2021. So I've been here for just over 3 years now. And I'm also an assistant professor at SFU in the Faculty of Health Science. And so I know you guys are a bioinformatics group, and I'm going to start with saying I am not a bioinformatician. I would say I'm trained primarily as an integrative exercise physiologist, but I did a PhD in innate immunity and molecular biology. And so I try to merge those two things together as best I can. And so today's talk is going to kind of bring in some legacy data from when I did my training my PhD and postdoc, some data that we have that's being published soon, and then at the end, I'll talk about some of the projects we have ongoing in the lab here. The focus of my talk is focused on something called inflammatory memory and how we can use omics-based tools to illuminate new pathology, specifically in heart disease. Okay, so just before I start, I just want to acknowledge that I work on the traditional ancestral and unceded territory of the Coast Salish people, including the Musqueam, Squamish, and Tsleil-Waututh nations. And a nice graphic here of where those nations sit and overlap within the Lower Mainland. Okay, so I was asked to talk about an exciting concept or state-of-the-art concept in our research or in my research that we do in our lab. And so the concept that I thought would be interesting for this group and for those that do immunology or changes in chronic disease or even infectious disease, we think about inflammation a lot. And so this idea of inflammatory memory is one that's relatively new. I'd say, kind of the initial concepts of this came up in around the mid 2000s, got bolstered around 2014, 2015, and now we have this massive expansion of ideas of how this concept regulates the adaptation that we have to various stimuli in our environment, and then how that dictates responses to either concurrent or subsequent challenges as well. And so what was just very elegantly described in a much better way than I could do, classic immunological memory, we think about our adaptive immune systems, right? So we think about T cells and B cells. We have this naive pool of cells. They get exposed to an infection, right? We get clonal expansion. We get recombination of various genes to enable this specific memory response that can last for years within either specific T cell receptor clones or within antibodies that are formed from B cells, right? And so this is traditionally how we've always thought of memory in our immune system. But as I said, back in about 2007, and we had evidence of this before, but it was really the first time that we saw kind of the mechanisms behind this. Traditionally, textbook biology suggests that, our innate immune system is kind of our first responder to various infections or pathogens or perturbations in our system and inflammation that can happen to sterile events as well in the host, like, some type of, we're going to talk about heart attack, myocardial infarction. But traditionally, people have thought that cells like monocytes and macrophages, neutrophils, they respond to cues from an inflammatory stimulus. They arrive, they exert their effects based on the environment that they get exposed to, and then they go back to normal, right? And cells just come and they're this kind of non-specific response cell, right? And so this is, a cutoff version of a figure I'm about to show. But if we think about the strength of the innate immune response here, right? And then we think about, this primary stimulation. And as I said, that could be a bug, that could be a heart attack, that could be anything. We think of this initial response and the resolution of that response, right? Especially if we are able to resolve, the infection or the tissue damage, what have you. But as I said, over the last 2 decades, what we've realized is that primary stimulus, and depending on what that primary stimulus is, can actually induce altered responses to a secondary stimulus. And that secondary stimulus could come days, weeks, or months after the resolution of that first primary stimulus. And this has been termed two different things based on two different responses. One is considered trained immunity, which was brought in by a group, or the term was brought in by someone by the name of Natia, and their first paper in Science on this kind of came in 2014. And it was actually adapted from plant biology, where they realized that even cells that don't have this recombination ability, like we see in T cells or an adaptive immune system, that innate immune cells can actually have this primed response. And so one of the key primary stimuli that we found that induced this is something like the BCG vaccine, this 100-year-old vaccine for tuberculosis. We also know that beta-glucan, which is this fungal wall constituent, can induce this response. And essentially what you see is if you give a primary stimulus of beta-glucan or BCG vaccine and you allow the system to normalize, come back down to baseline or homeostasis, when you hit those cells or hit that system with another stimulus, that could be endotoxin, LPS, what have you, you get this exaggerated response. On the flip side of that, we see that some stimuli can induce tolerance, which, unlike this potentiated response in trained immunity, we actually see no response or a blunted response. And this has been shown primarily with endotoxin, which is a component of the outer membrane of gram-negative bacteria or E. coli. Most of us think of it as LPS or lipopolysaccharides. So when you dose specific doses of endotoxin, you can actually blunt these responses of these cells. And so, as I said, people have seen these responses for a while. The mechanisms behind them are now coming out in droves. And I'm going to talk specifically about one of these today, but we can, and how it affects some of the others. But there's a lot that changes within these cells to induce this specific phenotype. The main one is epigenetic modifications. So we actually see changes to the shape of the chromatin from that primary stimulus that either primes or blunts that response. And that's based on the opening or closing of transcription factor or areas for transcription factor binding, enhancers, promoters, etc. We see reprogramming of these cells' metabolism, so they, are able to activate glycolysis to a greater degree to induce these quicker responses or shift towards the TCA cycle. It's a balance between each depending on the stimulus. But the main part is, we see altered responsiveness, right? Either this potentiated response or this blunted response, depending on training and tolerance. And these effects appear to be long-term, suggesting that particularly in monocytes and macrophages, monocytes in particular that only live about 5 days, it's likely also happening in the progenitors that make these cells, which is in the bone marrow and sometimes in the spleen. And so this is the concept I thought, during my training was super cool, kind of expands this idea of how our system adapts. And what we know now, originally, as I said, back in 2014, let's say, we started understanding this in monocytes, but we now know that these different stimuli, whether it be microbial, inflammatory, metabolic, right, that they can induce changes in a number of different cell types. So not just monocytes and macrophages, but also in epithelia, innate lymphoid cells, endothelial cells, NK cells, stem cells, hematopoietic stem and progenitor cells, tissue macrophages, microglia in the brain. And when you remove those stimuli, these cells all remain in this prime state. You add a secondary stimulus, and not only do we see inflammation go up through cytokines, but we see antimicrobial responses, chemokines to attract other cells in, proliferation, migration of these cells. And as you can imagine, this response can sometimes be good and sometimes not be so good. And it's being kind of written out in terms of, from a physiologic perspective, it defends us from pathogens. It might enhance wound repair, but in certain inflammatory diseases or in cancer, it can be pathologic. We're doing a lot of work in these different diseases outside of heart disease, which I'm going to talk about today. But as you can imagine, as an overarching construct, this inflammatory memory is really, I think, and, I've spoken to Shruti Naik, who wrote this paper a few years ago, and she's been kind of the proponent of this because she found a lot of these changes in epithelial cells. is that this is really, I think, a fundamental way our bodies adapt to our environment. And I think it's a critical component to think about in terms of disease pathogenesis and, mixing and matching these stimuli and what we're exposed to initially is likely going to then dictate how we respond to things in the future. Okay, so that's the overall concept. So now what I'm going to do is I'm going to go into this in the setting of CVD or cardiovascular disease or heart disease. And I do this because, again, this is kind of where my thinking on this concept started, and we started with monocytes, but we're moving into some of these other cell types as well. Okay, so let's think about heart disease, right? The thing that generally, or one of the main things that that people succumb to heart disease with is a cardiac event. Myocardial infarction, heart attack is one of the primary cardiac events that individuals will experience who have underlying coronary artery disease or cardiovascular disease. And so a cardiac event like a heart attack is an acute inflammatory, sterile inflammatory event, right? So you get this massive inflammatory hit without a pathogen there, right? And it activates the innate immune system, just like we see with the pathogen, and this disrupts overall, systemic homeostasis. And so this was from a review a few years ago, but it explains kind of the overall systemic or disruption of systemic homeostasis that happens with a heart attack. So if you start here, right, you have this heart attack, which is where you have a thrombus form in a coronary artery, which can occlude blood flow and lead to tissue necrosis of the left ventricle. And what that does is it causes this pain, anxiety, alarm and danger signal response that catapults the central nervous system and sympathetics to activate. It also activates beta-3 agonists and these, alarm or danger signals like IL-1 beta to signal to the bone marrow. The bone marrow then says, we've got to go and fix that damaged tissue. So it sends progenitors, leukocyte progenitors out into the spleen, this thing called extramedullary hematopoiesis. But the main effect is it dumps a whole bunch of immune cells or innate immune cells, pro-inflammatory monocytes, into the circulation. Now, that dumping of monocytes is integral for those cells to go back and repair the heart, right? Those are the cells that are principally going to eat up that dead tissue, signal other immune cells to come in, and then facilitate the repair or the formation of a scar. This idea of systemic homeostasis or driving this response, driving progression of cardiovascular disease independent of the tissue, is also that these cells get recruited to underlying atherosclerotic plaques, right? These are where the clot formation will happen when these things become unstable in the coronary artery. They'll shear off, lead to the thrombus formation, which can block the coronary artery and cause the heart attack. And so, Peter Libby, Matthias Nerendorf, these guys, they all found that these cells can go, you get more numbers coming out from the heart attack and drives this response. But this was under the assumption, right, and is under the assumption that these cells are nonspecific. They come out, they go, they fix, they're done, right? And so the question that we've had is, does something like MI, a cardiac event, does that induce this inflammatory memory, right? Do we see changes in these cells? Do the epigenomes of these cells change in such a way that might actually alter their function as well? And not only during that event, how does that function change, but after the event is what we were really interested in. So individual recovers, we see that now they have the scar tissue formed. Is their immune system still operating the way that it should? Okay. And people have looked at in terms of, these cells are activated transcriptionally. They have heightened inflammatory responses to respond to the heart attack, but no one had really looked at it long term. And so we've looked at this, and I'm going to share some data that's going to be published soon, kind of looking at the basics of this in terms of MI itself affecting this inflammatory memory phenotype. And so the basic construct of this is, if you think back to that first slide where we talked about a first stimulus and a second stimulus, we wanted to recapitulate this in a way that made clinical relevance for a patient who has a heart attack. So what we did was we used a mouse model system. So we induced an MI in mice where we ligate, so we actually tie off the coronary artery in these mice. and we induce a myocardial infarction. So we restrict blood flow to a certain part of the ventricle and induce a heart attack. And that induces a massive pro-inflammatory response, which is this line or small hump that we're representing here, right? And over about 10 days, it's pretty well established in mice and, within a few weeks in humans that pro-inflammatory response resolves and things come back down to that homeostatic level. So what we did was we had this heart attack, we waited 10 days in our mice, and then we hit them with a second stimulus. So we wanted to know, and we used a bacterial challenge. One of the main reasons that patients come back into the hospital after a heart attack is for an infection. And so we thought, well, let's hit them with a bacterial challenge and get a sense of how these cells respond. And if there's any kind of, molecular or cellular changes that happen. And the idea was, what are we going to see? Are we going to see that these cells are blunted? Are they going to be tolerant, like endotoxin tolerance? Or are we going to see this potentiated response, this trained immune response? To get at this first, what we wanted to do, as I said, we looked at, we wanted to look at, the overall structure of these cells. So we know that the epigenome is related to this memory. So we wanted to say, well, okay, is the epigenome changing in these cells? And so what we used was a technique called ATAC sequencing, which is a assay for transposase accessible chromatin. And what ATAC-seq does, it's quite clever. So when you think of epigenetics, we have DNA methylation. We can use ChIP-seq to look at, specific histone marks, various different chromatin marks that are associated with open or closed or activated or inactivated areas of DNA. What ATAC-seq does is it's an unbiased way to go at this, and how it does that is, our DNA is wrapped up in histones, and we have areas of open and closed chromatin, which is shown in this little figure here. And what I'll do here. So what ATAC-seq does is you use these transposomes that are essentially used to cut the areas of open chromatin at the base of that closed chromatin at both ends. And what you're able to do then is you're able to amplify those regions that are accessible and sequence them. And so you are able to essentially say which regions are more accessible and which regions are less accessible in a given scenario. And so our scenario was a heart attack or a mouse that had had a sham surgery. And we wanted to look and see, is that chromatin accessibility different, right? Vis-a-vis, is the epigenome of these cells different? And so we did this in monocytes in the bone marrow. That's where these monocytes are produced. What we found was there's about 800 differentially accessible regions in our analysis in a mouse that had had a heart attack versus that had had a sham. And remember, this is 10 days after. So heart attack is resolved. The tissue now has a scar, and we see these long-term changes. We use a number of different tools. As a biologist, I'm very interested in understanding the overall pathways or functional changes that are happening within these cells. And so we use something called GREAT, which is a genomic region enrichment of annotation tool, which essentially is a functional enrichment analysis that's directly performed on genomic regions. So it's gene independent, it's genomic region dependent. And what we did was, because we had a lot of regions that were more accessible in this MI scenario, is we looked at those genes that were open. And what we found was a number of interesting things: abnormal blood cell morphology, abnormal or increased susceptibility to infection, particularly bacterial infection, phagocyte morphology, abnormal cytokine levels, decreased cell migration. When we took the areas of open chromatin or more accessible chromatin and took the genes that were associated with that and ran gene ontology, we found, prediction activation for integrin median signaling, which is involved in migration and phagocytosis, reductions in cytokines like IL-6, regulation of phagocytosis down. So all these functions that monocytes are supposed to do, right? Eat bacteria, induce cytokine responses, migrate to, various stimuli. They seem bad at all, or seem as though their chromatin or the regions that are accessible are potentially making a landscape that would make them worse at these functions. So that was kind of cool. So then we were like, okay, well, let's see if they actually are worse at these functions. And so what we did here, using the same stimuli, is now we went in vitro. So we took these monocytes after a heart attack, and we hit them or tasked them, let's say, with doing different functional jobs. And so what we found was, if we took the monocytes, put them in a dish from a heart attack mouse versus a sham mouse, and we put GFP positive bacteria, dead bacteria on top of them, and asked them to phagocytose that bacteria, the MI mice were worse, or MI monocytes were worse at doing that. If we asked them to produce cytokines, while these weren't significant, we saw this downregulation of key inflammatory and anti-inflammatory cytokines. When we asked them to migrate to CCL2, which is a chemokine that attracts monocytes, they were worse at it. And when we put them in a well with live bacteria and simply asked who survives and who doesn't, the Staph aureus won. So we used staph here. We did similar things with E. coli. I'm not showing it. And the number of monocytes after a period of time, about 2 hours, that had died was significantly or nearly significantly there too. So overall, these cells become, I would argue, tolerant or somewhat dysfunctional after a heart attack. So that's not great, right? We obviously don't want to have an immune system or an innate immune system that isn't responding the way it should. Interestingly, what we did to find out if this was long lasting is we took bone marrow from mice that had had a heart attack, and we injected it into a lethally irradiated mouse. So did a bone marrow transplant, and then allowed these mice to recover over a period of time, so 4 weeks. And then we took the monocytes out of that bone marrow after the bone marrow had been reconstituted, and again, asked if these cells could do these tasks. And as you can see, their ability to phagocytose or migrate maintained this dysfunctional state. So suggesting that this may not only be happening in the monocytes, but also upstream in their progenitors in the bone marrow. Okay, to close this loop, right? So mice in vitro, or their monocytes in vitro, don't respond as well to perform these tasks. Their epigenomes show that they are epigenetically shifted to potentially support this dysfunction. And it's transferred by bone marrow. If we actually infect these mice in vivo with a bacteria like Staph aureus, these mice also don't survive. So in mice that had had a heart attack, recovered from their heart attack, and then we give them an intraperitoneal injection of Staph aureus, we see that the mice that had had the heart attack succumb to the infection much faster, or to a greater degree. They lose more weight. And in the blood, at 4 hours, we don't see a difference, but at 18 hours across organs and systemically, we see an increased amount of bacteria. So the infection is essentially winning. So what this says from a heart attack perspective and inflammatory memory is it seems like a heart attack does induce this memory, more reminiscent of this tolerance concept. And it induces it through these learned epigenomic responses, driving deleterious effects to subsequent non-CVD sequelae or subsequent challenges in the system. Okay, so that's very interesting. And so these data, as I said, those were unpublished. I'm now going to go to a story that is published and was published a few years ago, but I think builds on this concept of memory and effects that it may have. So if we know after a heart attack that pro-inflammatory monocytes have this kind of tolerant response, their epigenetic modifications are changed, their responsiveness is different, we see these long-term effects sustaining. We did do metabolic work, but we didn't show it. We did show this in humans as well, which I didn't show, just for simplicity's sake. But so the question here is now, people who have a heart attack, they don't usually only have cardiovascular disease. Most of these patients will have other diseases or comorbidities happening at the same time, right? Concurrent or underlying comorbidities. So what happens in a population that has another disease, which we know can maybe impact this response, and then they have this heart attack as well? And, the world that I came from before moving to Vancouver was the world of cancer. And so I particularly worked in this area called cardio-oncology, which essentially is, cancer patients at a cancer diagnosis. And we're looking at this in the setting of breast cancer, but this is true for other cancers as well. Breast cancer patients get treated with a number of therapies that can be very good at killing cancer cells, but are also very good at killing other cells in the body. And things like radiation, chemotherapy, endocrine therapy can directly impact cardiovascular health, killing cardiomyocytes, altering vascular function, and overall increasing risk of cardiovascular disease and cardiac events. So these are patients that have a high risk of having a heart attack, but they have underlying cancer at the same time. And there's other indirect effects like age, inactivity, et cetera, that can drive this risk too. So we asked this question, is there cross-disease communication? And these patients that have underlying cancer, do we or have survived their cancer but maybe have residual non-detectable disease? Does a heart attack change the trajectory of their underlying malignancy? And I could go into this over a long period of time, but I'm going to do it very short. The answer is yes, and all this data is published if you want to take a look. We looked at it in patients, and so we looked at patients who had been diagnosed with primary breast cancer, so early stage primary breast cancer. And then in this cohort where we looked at them over a 20-year period, we used a group from Kaiser Permanente down in California to help us with this. We showed that individuals that have a cardiac event after their breast cancer diagnosis have a 60% increased risk of their cancer spreading / coming back, as well as dying from their cancer, which is really unfortunate, but says that there's probably some systemic driver of the MI or of the cardiac event that would be inducing a change to the underlying malignancy. And so to answer that, we developed a mouse model again. And so what we did here was we took mice that we induced a heart attack or a sham surgery, and we induced it in mice that had an implanted tumor in their mammary fat pad or spontaneously developed tumors in their fat pads. And what we found was if the mouse had had a heart attack or not, we saw this acceleration of tumor growth in both models, as well as an increase in the metastases to the lung. So in cancer, it usually starts at a primary site, and then you get movement or metastases of that primary site to distant organs, and that's where we get into this incurable or the disseminated state. And so in our mice, it kind of recapitulated what we saw in our patients, where we have this increased risk of progression, right? But we see it, as essentially a causal factor here in driving this response in our mice. Okay, so the question then is, how is it doing this? And I've given away the answer, but we wanted to look at this inflammatory memory in this setting, right? And we did a number of assays, which I'm not showing, but I'm just going to focus on the bioinformatic piece because I think it's quite interesting. Specifically, again, here we ran ATAC-seq, and we did this back in the bone marrow, just as we did before. But now, remember, we aren't just looking at MI alone by itself, but we're looking at it in the context of cancer, right? And we know that tumors have a massive impact on hematopoiesis and the bone marrow composition. And so we saw in this combination state a unique signature within the chromatin landscape of these cells, because they're getting signals from the tumor, they're getting signals from the MI, and they're likely getting signals from both. Again, when we looked at using GREAT analysis, and within GREAT you can use gene ontology, when we look at the genes that are now less accessible, which is unique. Remember when I said in MI alone we had more that were, majority of genes were more accessible. But if you look at less accessible loci, we see this enhanced immune suppressive response. So again, this is just the chromatin landscape that's been primed, right? And we see downregulation of cytokine production, lymphocyte activation. One of the big things that monocytes need to do is they antigen present to T cells. So their ability to present seem to be, or activate these cells seem to be a little bit perturbed. And when we looked at transcription factor binding sites, or enrichment for transcription factor binding sites. We found a multiple of interesting ones, but we found these pioneering factors for monocyte differentiation, PU1 and SPIB, CEPB, were enriched in these less accessible regions. Those are important for monocytes to turn into macrophages, which is the kind of natural progression of those cells. When they get hung up, that can be indicative of this immunosuppressive signature in these types of cells. We also saw IRF8, and IRF8 is an interferon response factor, which binds with PU1 at monocyte enhancers to activate inflammatory responses. So lots of interesting hits. So that's changes in the chromatin in the bone marrow. We wanted to know, do those changes in chromatin in the bone marrow impact the responses of these cells inside the tissue of interest, which in this scenario is the tumor. And so we took out monocytes from the tumor because they got recruited there, and we showed that they were recruited to a higher degree as well. But when we looked at RNA sequencing of these cells, so now we're looking at gene expression changes, we found a number of genes that were differentially expressed, some up and some down. And when we looked at genes separated as downregulated or upregulated, we saw similar features to what we saw in the gene expression or in the chromatin accessibility in the bone marrow. So we see again, inactivation or downregulation of genes related to lymphocyte activation, adaptive immune system, interferon gamma signaling, so inflammatory responses. We didn't see anything massively interesting on the upregulated side. We use a program called Ingenuity Pathway Analysis in our lab a lot. And this is a really great program because unlike GSEA, where you kind of have to separate out up or down regulated genes, IPA can take all of those genes in together, and then it takes into account fold change and p-value or p-adjusted value to predict activation or inhibition based on a large database that they have based on other studies that can give you an indication of, what's changing. And so this analysis is for looking at upstream regulators. So it's not that these regulators that are on the y-axis here are changing, it's that the genes that regulate those factors are changing. And what you can see is we have predicted inhibition of, major inflammatory drivers like interferon gamma and STAT1, TLRs as well. And we see activation of anti-inflammatory factors like the IL-10 receptor. Okay, what we're doing more in our lab now, and this was kind of the initial way of doing it, was we wanted to know, okay, so we see these changes in the bone marrow, we see in the chromatin in the bone marrow, we see these changes transcriptionally in the tumor. How do they connect? Can we integrate these data sets? And we did this in a very preliminary way. But what we found was that genes that were transcriptionally inhibited in tumor monocytes, I'm calling them MDSCs here because they're these monocyte-derived suppressor cells, but in these monocytes in the tumor, genes were inhibited that also exhibited reduced chromatin accessibility back up in the bone marrow. So they're produced in the bone marrow. The idea here is that their chromatin is less accessible, and when they enter into the tumor, that reduced chromatin accessibility appears to be decreasing their gene expression. And we saw this across a number of different genes related to specific transcription factors that we knew are related to this immunosuppression, right? So interferon response factors, pioneering factors like PU1 and CEBP. Some of the main genes that we saw were things like CD40 and CD86. These are very important in T cell activation. I'm not showing the data here, but when we put these monocytes in a co-culture with T cells and activated the T cells, the monocytes from MI-induced mice were able to, or they suppressed T cell activation compared to the sham, validating this at the functional level as well. CLEC7A is it's the dectin-1, it's the gene product for dectin-1, which is involved in innate immune memory in response to BCG vaccine and all those things as well. So this training response, these genes that are needed for the training response or the potentiated response of cells, is actually here. The transcription is down and chromatin accessibility is down, maybe part of this tolerance type phenotype. IRF8 itself, as well as I said before, that was a gene or a transcription factor that's needed for differentiation of these cells into macrophages and to function appropriately. Okay, one of the other things we did, and again, we're working with our graduate team now and bioinformaticians like you guys to figure out better ways to do this, but this was our way of doing it initially back, a few years ago. But what we did was we also had RNA sequencing in the blood. So what we're doing here now, remember I said there's gene or transcriptional changes in the bone marrow or in the tumor. Did we also see those transcriptional changes happening in the blood? Are there systemic changes happening in the blood monocytes driven from the bone marrow? So chromatin accessibility change in the bone marrow, transcriptional response in the blood, transcriptional response in the tumor. And what this analysis did was we took the top 1,000 differentially expressed genes in the tumor, and we simply asked those that are upregulated in MI or those that are downregulated in MI, are they also enriched in the Lysic C high monocytes in the blood? And the answer was yes. Significantly as a group, we found that these genes shifted. So where we didn't have this robust group of genes that were changing, we did see that the phenotype from, this was another piece of evidence to suggest that the chromatin changes in the bone marrow affect how these cells operate at a gene expression level in the blood and also change how they are expressed or function in the tumor. Okay, so that's the overall concept here. And just kind of a summary of that put into the context of now what we know about inflammatory memory is that something like acute myocardial infarction is another one of those primary stimuli, right? And what we know is that that changes monocytes in the bone marrow. We didn't specifically look at it, but based on the fact that we can extend these in bone marrow transplants suggests that we are absolutely also, or MI is absolutely also changing hematopoietic stem cells. So we have these varying different progenitor populations in the bone marrow, from its most stem-like state all the way down into its differentiated monocyte state. And this is a concept known as central memory. So when we're thinking about monocytes, people think about, okay, are these changes happening centrally in the main production sites? And what we showed, as I said, was that this MI drives these changes epigenetically. These cells then go into the bloodstream when tasked with a secondary challenge. In this case, it was cancer, or we also showed it with infection. These cells then travel to the periphery, right? Either in the blood or into tissues. And what we showed was systemically in the blood, right, to an infection, that these changes in the bone marrow drive changes in the secondary responses, this learned behavior. But it also, within a tissue like the tumor, can drive tumor growth, right? Where we see changes in transcriptional, gene expression changes, and functional changes. Going forward in this world of, specific to MI, we know that there's this systemic effect, and monocytes are not the only cell that are being affected, recruited, or being utilized in this systemic response to a heart attack, right? We know that there are other cell types involved, either independent, like in the setting of MI alone, or in the setting of response to infection, or in this response to an underlying malignancy. So cells like NK cells, innate lymphoid cells, dendritic cells, just to name a few. What we also know, and I mentioned this off the beginning, is that this is not exclusive to monocytes, right? Smooth muscle cells that wrap the coronary arteries, smooth muscle cells that wrap all of our blood vessels, epithelial stem cells. We know that after a heart attack, you get this increase of perfusion of liquid and fluid into the lungs, which can then drive changes in epithelial cells. We know that fibroblasts, they're recruited to fix the scar. They're going to be impacted, right? Or stromal stem cells, which then differentiate into those fibroblasts. So it's a very complex story that we're only starting to understand, but I think it's a really interesting one when we try to frame biology within the context of an ever aging human that everyone has had these unique exposures. It's really going to drive a complex pattern of response and unique pattern of response, which we can use these omics tools to better understand and articulate. So for us, what are we doing now? So again, we've done a lot of this in heart disease, and that's what I've described, but we're interested in this across both other pathologic states and physiologic states. And so I'll explain those shortly here. So again, this was the slide I showed at the beginning, but these different stimuli, right? Whether it be a bug, whether it be sterile inflammation, whether it be metabolic, people have shown that high-fat diet can induce a lot of these changes. And studies are coming out every week of like, different primary stimuli that affect either short-lived or long-lived cells. Removal of that stimulus leaves these cells in a prime state. Pick your poison of that secondary stimulus, have these effects, and drive responses. I think what's unique for us is we really want to understand how these things all operate together. And so we're utilizing a few newer techniques that'll really allow us to do this in a better way. And so, what we've done previously is bulk ATAC-seq, so 50,000 monocytes altogether, but we know that those monocytes are all going to be impacted in a slightly different way. And there's different subpopulations likely that would be created based on these stimuli. So the epigenome will be different. We also know that, as showing on the left, there's lots of different cells that are doing this. So we're using single cell ATAC-seq along with single cell RNA-seq, and now combining or integrating those within blood and tissue in various states. So pathologic states, one of the things that we're really interested in is a disease called COPD, or chronic obstructive pulmonary disease. This disease has a newfound appreciation for myeloid cells like macrophages in driving disease pathology. We also know that epithelial cells get very dysregulated in the state of COPD. COPD is where you have essentially obstructive disease because your small airways become floppy and collapse. And so you end up trapping gas in your lungs and leads to these things called exacerbations and high risk of infection and all this other stuff. So we're working with clinicians at St. Paul's to define this process, not only in monocytes and macrophages in the lung, but in monocytes in the blood. We're even isolating hematopoietic stem cells in the blood to try to get at these states, and then try to look at them across that central to peripheral continuum to see how things are changing centrally, what's changing peripherally. We're also doing things on the flip side of that. So in a physiologic state, as I mentioned at the very beginning, I'm trained as an exercise physiologist. So we've got a few grants and a number of grad students working on projects where we're trying to understand, if these pathologic stimuli can drive these deleterious changes, what about something that's physiologic, something that improves immune responses like exercise? Does it change the epigenome? Does it induce this exercise-specific memory that may allow us to understand how exercise protects us from these innate immune or immune-related diseases? So with that, I'll just say thanks to everyone that helped with this. Again, my lab has a number of incredibly strong students, and I think some of them are even on the call right now. We've got Sarah, who's at SFU and FHS. She's working on a number of projects in sepsis, and can we reverse some of the tolerant phenotypes we see in sepsis, these immune-tolerant phenotypes. Malad, who's doing the work in COPD and also some exercise work. Ali doing the exercise work. Josie as well. Paneet working in community-acquired pneumonia, but using all the concepts that we just described. Janelle is our research manager. She runs a lot of our clinical trials. She and Jeffrey are undergrads who have helped with these projects as well. So with that, I'm happy to take any questions you guys have, and yeah, hopefully I can answer them.
Thank you, Dr. Graeme Koelwyn. That was really good talk. I really appreciate it. I have a question about the paper that you published previously about the cancer and heart attack and their correlation. So I was wondering what type of cancer type was it?
Yeah, so
Like,
what we showed was a group of breast cancer patients. So it was, we didn't subdivide by tumor subtype within breast cancer. So we had all breast cancer comers. And it was individuals who had been diagnosed with primary breast cancer, so stage 1, stage 2 disease. And we were looking at least 6 months after their primary diagnosis if they'd had that infarct. So that was the group that we looked at there. But other groups since this point, this is, there's this resurgence of this idea of, we call it cardio-oncology. So people have always thought about how cancer treatments cause heart disease. People are now calling it reverse cardio-oncology, where, the heart disease causes the cancer. And so across a number of different cancer subtypes, they're showing that heart disease can increase the incidence of disease and progression, not just breast cancer. But our data was breast cancer specific.
Oh, so you're hypothesizing that it will be like general trend across all cancer types, not just breast cancer specific.
I think it could be. I mean, the world of oncology is very unique in the fact that different tumors with different clonal dynamics or mutational burdens are going to respond to different factors in a unique way. So it's
Mm-hmm.
probably, if I'm hedging my bets, it's probably less to do, location is going to matter. But I think probably mutational profile is going to also have a major effect in terms of, what's the gene environment interaction happening within those subtypes. I think that's, we think about this on the exercise side of things too, right? Exercise restricts tumor or reduces the risk of both incidence and progression of disease, but it's likely specific to, the type of cancer and the competitive advantage that certain cancer clones would have over others. But we're still years away from trying to figure some of that stuff out, at least on the
Mm-hmm.
cardiovascular or exercise side.
Mm-hmm. I see. Well, that's interesting. Okay, if you have any questions in the audience, feel free to raise your hand or just voice it out directly. If we don't have more questions, I'll just go on with the different questions. So like, it seems like you ran a lot of pathoanalysis enrichment um, for like significant result for significant result, but like, do you think that so like also you use like many different pathoanalysis tools like Ingenuity, IPA, like great, but like is there any like specific reason why I chose different pathoanalysis tool for each pathoanalysis?
Yeah, that's a good question. We take the technique of using all of them and showing
Mhm,
that we have biological redundancy. I think that generally helps when we publish, if we can show that it happens across multiple different database sets. So we
mhm,
found success that way. We've also found success in, I showed, all of the different enrichments that we had, but we validated those with assays that showed that they were functionally relevant, right? Because as we all
mhm,
know, these things are not perfect and they give you a lot of false positives or,
mhm,
biology that may not be fitting within your own construct, right? So I think we
mhm,
find it as a way to give us, as many people do, a readout, right? We try to see which
mhm,
ones do we get consistently showing across these different things, and then we go and validate
mhm,
the features that we see are most relevant, right? And it doesn't work all
mhm,
the time,
mhm,
but it definitely helps. And I think that especially when you're working in kind of translational or clinical spaces, I think a lot of
mhm,
people that operate further on the clinical side really like to see that relevance, right?
mhm,
So those systems are very good to help us kind of steer the ship towards where we want to go. And a lot of what we do is, as I showed, is less, single gene specific phenotypes and more overall functional changes. And so it works for us, but it obviously, it comes with limitations that we know. And that's why we try
mhm,
to use a lot of them to help us better
mhm.
articulate what's happening.
All right. Um, and also like you mentioned that like you're trying to like you're so basically you're studying epigenetic signatures learned by heart attack. Like I was wondering like do you have any like specific epigenetic signatures of your interest or your significance? Because like there are many epigenetic signatures, so yeah, I was wondering what's your
So in
like
terms of
interest.
what type of epigenetic analysis
Mm-hmm.
you like to do? Yeah, I mean, as I said, I mean, we take what we can get. So, we've got one data set where we've got access to over 1,000 individuals with methylation-seq, right? So we're
Mm-hmm.
using that to help us
Mm-hmm.
understand, hyper and hypomethylation, roughly
Mm-hmm.
correlate with, activation or inhibition.
Mm-hmm.
I do like ATAC-seq because it's a low cell input, which helps on our
Mm-hmm.
human trials and helps on our mouse stuff as well. But I like this, agnostic component to it, right? Because to me, it gives me this kind of, true landscape of like what's open or closed. Now, what's open doesn't mean it's necessarily going to be transcribed, right? But it at least
Mm-hmm.
gives us a sense of what's happening. And so I found it to be an incredibly effective tool to
Mm-hmm.
understand these epigenetics, because when you run methylation or you run acetylation, you look for specific histone marks. Um, I see that as almost that's like a secondary validation, right? Where you really
Mm-hmm.
want to know which marks could we look at, or if you're trying to develop prognostic tools or things like that. So yeah, so that's kind of where I start. But again, I'm not an epigeneticist, right?
Mm-hmm.
I'm a biologist, and so to me, I like that because it gives me this kind of broader overview. But
Mm-hmm.
obviously, you could use other techniques to get it more specific, chromatin
I
specific.
see. Mm-hmm. I see. We have one question about epigenetics in the chat. I'll voice it for you. So Robert Bell asked, how do you think the signal of MI travels to the bone marrow to change epigenetics, and could that signal be blocked?
Yeah, great question. Um, we don't know. I have a feeling it has to do, again, as I say, like I think this is a biologically redundant, I believe it to be a biologically redundant response to acute inflammatory signaling, right? So when you think about, well, what are the signals of that, right?
Mm-hmm.
I would say that's autonomic, sympathetic activation, right? That's going to be that kind of um, alarmin type response, right? I think all of those are inducing that activation. Is it the simple requirement of proliferation of those stem cells that could also be driving that response? I think all of those things are on the table. And then the question about could it be blocked is a great one. The hard part with these is you need the response to fix the infarct, right? Now, you can have a hyperactive response, which you could maybe try to dial down. But if you think of it as like a U-shaped relationship, right? Over response, not good. Middle response, good. Not enough response, not good, right? In terms of, myeloid activation, release, and entry into the heart attack. So, I think it's probably more about can you balance or ensure that initial inflammatory response is appropriate. And that gets into what has the individual prophylactically been exposed to prior to that infarct, right? So someone like me, someone who's highly fit, they probably can dose that response better or resolve that response better, which may then limit that secondary immunosuppression, right? Return to homeostasis faster than someone who is maybe in a hyperinflammatory state to begin with. Um, but I think, mechanistically testing, I think the way you could do it is you could block sympathetics, and then you could look and see if you restrict that memory. But you'll probably also then have effects on the MI specific outcome, so it's a bit of a dam to think about.
Great. Do we have any questions in the audience? Feel free to raise your hand if you have one question. Um, I have a simple question. So like you are planning to use single cell
Mhm.
ATAC-seq in the future, or is it ongoing, or is it like still on the plan, or
Yeah, so we've got it's moving now. So yeah, so we, single cell comes with a few additional complications, right? We don't have the same depth to be able to understand some of these changes. And so time will tell what we're really able to see. We're going to be able to pull out, the stronger phenotypes, I suppose, or the changes that are more available, I suppose. But I think the breadth is what's going to give us some really nice information. I mean, the one thing that we have done in this, and we've spoken to a few groups about it, is, 10x offers this multi-ohm package, right? So you can actually do the two of them together. And what I've heard from a number of colleagues is, if you're doing in the same cell, single cell ATAC-seq and single cell RNA-seq, you are restricted on your RNA-seq side to nuclear RNA. And so, that could be anywhere between, probably around, 10% of the actual RNAs that are there. Most of them are going to be out in the cytoplasm or other places, right? So we've decided to do them separately and integrate, which I think will give us better alignment of the two. But again, we're working through, I mean, Robert is probably going to be involved in this because we're working with the Prostate Center on this. To figure out how we can best integrate those and get the coverage that we want. So kind of balance that breadth versus depth and integration appropriately. So it's not for the faint of heart, but I think we've got at least an initial plan of attack to try that.
I see. Yeah, well, overall, I was really lesson two talk is really shocking that actually heart attack really does its like effect on memory and also like immunity. So like that was really shocking and interesting. Um, yeah, it was really interesting talk. And do we have any questions in the audience? If we don't, will it be okay to close the seminar today? Cordia and Madeline, Jonathan? Do you have any
Yep.
announcement?
Definitely. And for everyone who's here, thank you for joining us. Just at the end here, we want to share a survey with the Canadian Bioinformatics Hub. They are looking to determine some topics, formats, and locations for training programs in 2025 in the new year. If anyone's interested and feels like, this is something they could benefit from, I'm just going to put a link to their surveys in the chat. And thank you all. Thank you to the speakers as well. That was really interesting. But yeah.
Great. Thanks, everybody.
Thanks for joining.